Work? I guess not. Okay. Thank you. Sorry for our IT challenge at this point. Thank you so much for the patience and thank you so much for attending this session here. This is a replacement workshop for another one that was canceled. So this is a last minute thing. I'm very thankful that you still make it to this workshop. And I cannot play my slides from this computer. Our IT gentleman will play from his computer. I just emailed the slides to him. And in the meantime, if you don't mind, I welcome you to log into a Zoom. And if you have any questions, you can type your question in the Zoom chat box, and we have Alex there to answer your questions if you have any. So the Zoom login ID is, if you don't mind, write down 993 9408 9082. And I'll say that again, 993 9408 9082. Thank you so much. And in the room chat box, I just sent a Google Drive link. And in that Google Drive, you will find everything you will need for our hands-on session today. Yeah. It's 993 9408 9082. Alex, can you hear me? So we have Dr. Alex Smirnov on Zoom. And if you have any questions, you can type your question in the chat box. And hopefully he can answer your questions where I go through the slides. On Windows, I don't know how to make it a full screen. Okay, I guess that's full screen. Okay.
We do. There we go. Try clicking through, see if that works.
No.
You might have to do
Page down. No,
page.
page down.
There we go. Ooh, I hit too many buttons. There we go. And page down. There you go.
Thank you so
I might just have to scroll a little bit more, but yeah, page down.
much. So, are you able to access a Google Drive folder? Oh, good. And you might try to download the raw data files while we talk. It might take a few minutes for you to download. Okay. So this workshop is about the informatics tools that my lab has developed for untargeted mass spectrometry-based metabolomics and exposomics. And the software framework is called ADAP. And ADAP stands for Automated Data Analysis Pipeline. Okay. So I'm sure all of you know this already, but just to make this slide self-contained, we have a few background slides to get us all on the same page. So human metabolome and exposome consists of many compounds, both endogenous and exogenous. And we know our human body produces lots of compounds. In addition to that, we are exposed to hundreds and thousands of chemicals as well. So metabolomics and exposomics try to identify and quantify, I should say semi-quantify compounds in our body. And both targeted and untargeted metabolomics and exposomics approach are used. And we all know there are two primary analytical techniques that are used for that purpose. One is AMR, another one is mass spec. And this workshop is about the informatics for mass spectrometry-based metabolomics and exposomics. Okay. So a typical workflow for untargeted metabolomics and exposomics using mass spectrometry consists of multiple steps. So conceptually, we need to have sample collection step first, and then sample preparation step after that. And then we analyze samples on mass spectrometry. And after that, we need to process the data. And after the data processing, we need to do biological interpretations. So today's workshop is about the data processing, which means to make sense of the compound signals in our mass spectrometry data. This includes gas chromatography mass spectrometry data and liquid chromatography mass spectrometry data. Okay. So as we all know, liquid chromatography and gas chromatography is to separate our complex samples to make our spectra not so complex, right? And so no matter it's liquid chromatography or gas chromatography, it gives us the dimension in retention time. And following that, our mass spectrometry serve as analyzers, and that will give two more dimensions in our data. One is mass to charge ratio, another is intensity. So our informatics tools start with dealing with these three-dimensional data sets. All the information about our compounds are encoded in this three-dimensional data set. So we have been developing an informatics framework for making sense of the LCMS or GCMS untargeted metabolomics and exposomics data. And this framework consists of multiple tools. ADAP-BIG is to pre-process our raw instrument LCMS and GCMS data. And after that, we have ADAP Analytics, which is a statistics and machine learning package. And after that, we have ADAP-KDB, and that's a cloud resource for identifying compounds. annotating compounds, and prioritizing unknown and known signals. And to interpret the compound signals that we have detected, we are developing MetaboFood, which links food compounds with metabolic pathways and diseases. And the final component is ADAP-Exposome, which is a resource we are developing for specifically for exposomics. And among these tools, ADAP-BIG is the only desktop software tool. All the other software tools are cloud-based. And today, we're only going to focus on ADAP-BIG and ADAP-KDB. And this is to extract compound signals and try to assign compound identities to the signals. Okay. So here you can see the web links for you to access these tools. Okay. So first, let's look at ADAP-BIG, which is a desktop software tool for pre-processing untargeted LCMS and GCMS metabolomics and exposomics samples. In particular, this ADAP-BIG was designed and implemented to handle large-scale studies where you might have thousands of samples. The development of the algorithms for ADAP-BIG actually have been ongoing for quite a number of years. We started with processing GC data and gradually and advanced into LCMS data as well. So that's an algorithm development. And in the meantime, we have been developing the software tools as well. So you can see we started with implementing those algorithms in C++ and R, and gradually advanced to Java. Right now, it's primarily Java-based. And in particular, about a few years ago, we started developing ADAP-BIG for processing large-scale studies. And I have a few pictures of those people who were involved, especially Alex, who was the mastermind behind the design of ADAP-BIG. And he is on Zoom if you have any questions for him. And the ADAP-BIG algorithms consist of many modules, of course. And this workflow that I'm showing here is especially for LCMS data processing. And today, our hands-on exercise will be primarily for LCMS data processing. And if you could use ADAP-BIG for LCMS data pre-processing, hopefully you'll have the basic knowledge for processing GCMS data. And here I listed some key people who contributed to this development. Okay. So within ADAP-BIG, currently there are 3 primary data processing workflows. One is for GCMS, one is for LCMS single batch. Basically, that means you only have one single analytical batch. And there is another workflow for multi-batch LC-MS data processing, where you might have thousands of samples to process. And we listed here individual steps for each workflow. And we'll go into a little bit into detail about these steps. Yes? Yeah, very good question. So this multi-batch processing is for studies where you have so many samples. You have to distribute those samples to many batches and to process them in the course of weeks or even months. Does that answer your question? Okay. So for GCMS workflow, the major steps include the first step is to extract chromatograms, and we all know extracted ion chromatograms is a chromatogram for a particular mass. And after that, we detect peaks in those extracted ion chromatograms, and then we try to purify the spectra by doing spectral deconvolution. This step is originally in MDIS, and we borrowed some ideas from that and implemented our own algorithms. And then align samples that are acquired at different time, and then we'll try to annotate those signals. And for LC-MS single batch processing, and there are some similar steps as processing GCMS data. That includes constructing EICs and peak detection. But there is a step that's distinctly different for pre-processing LC-MS data, and that's the MS-MS-MS pairing, which means we link those MS1 peaks with the MS-MS spectra. Okay? And then after that, we have alignment, and again, identifying and annotating our signals. I just realized I should have shared my screen so that Alex could see where we are. And in addition to the built-in workflows, users, when you use ADAP-BIG, you can also create your own customized workflows because we have some algorithms built into the tool so that you can create your own custom workflows. We are still working on this, of course, but in the near future, we'll make this more and more smooth. And these individual steps that you can use to build your customized workflows include ADAPT search. This is especially for LC-MS, of course. Background removal when you have blank runs. And batch effect correction and when you have multi-batches. And gap filling when you want to address your missing data in your processing. And also filtering data if you have study pools. Okay? And the customized workflows could be created and using XML files. You see here, do you see that? This is XML files that basically describe your customized workflows. And for multi-batch workflow, where you have many analytical batches acquired over the course of several months or even more than a year. And we have basically between batch processing and after within batch processing to handle the batch effects. And in addition, there is multiple places in that workflow to check whether or not the data meets the expectations. And this is primarily principal component analysis to see where the different types of samples, especially those quality control samples, are in your PCA scores plot. And here I included one example application just to give you an idea how fast ADAP-BIG is after we implemented those optimizations in our algorithm implementations. In this example applications, we have a total of 42 analytical batches. And from these 42 analytical batches, the total number of samples is about 4,400. And within each batch, there are 80 study samples, and there are 8 blank run samples to help us filter out those background noise peaks. And there are 8 NIST quality control samples, and also there are 8 study pools to help us further filtering out the data. And we run ADAP-BIG on a desktop computer with 16 CPU cores and 64 gig of RAM. It took a total about 30 hours to finish. Okay. And this development of this multi-batch processing was done in close collaboration with Professor Susan Sumner at UNC Chapel Hill. Now let's take a closer look at the ADAP-BIG pre-processing software. And this software has lots of data visualization capabilities to help you interact with the raw data, intermediate results, and the final results. And in order to make ADAP-BIG scalable and for processing large studies, and we had to use modularization and to release computer resources after a particular step is done. And in addition to that, there is an ADAP Analytics microservice-based tool that ADAP-BIG communicates with, and where we can do machine learning analysis after your data preprocessing. And the software is available on GitHub, and here is the URL where you could download ADAP-BIG for preprocessing. And we have different installation packages for your different operating systems. And no matter you have a Windows machine, Mac, or Linux, you can download the installation packages for your operating system. You can also run ADAP-BIG in console, and you can incorporate this into your computer cluster workflow if you have really large-scale studies. And we also have individual Java modules to give you even more flexibility. Okay? And here you can see the website, basically, ADAP-BIG github.io. So when you go to this GitHub webpage, you can see different installation packages. And we have many versions there. Right now, the newest version, which is not completely fully tested yet, but it's usable, and today we are going to use it. It is version 1.00, and you can see here the different packages and Java modules for you to use. Okay. So ADAP data preprocessing is also available in MZMine. So for those of you who are very comfortable with MZMine, you can use both the LCMS processing and the GCMS processing capability of ADAP in the MZMine framework, especially the GCMS processing capability. And on the MZMine website, there is a tutorial for you to follow to process your GCMS and LCMS data. So that's a conceptual description about ADAP-BIG data preprocessing. So after the preprocessing, you will get a peak table for semi-quantitation. And if you have LC, MSMS, or GCMS data, of course, you will get a spectra. And you want to know what compounds could have produced those spectra. That's where ADAP-KDB comes into the picture. And ADAP-KDB is a cloud resource for you to identify and annotating your signals, as well as for prioritizing and tracking both known and unknown signals. And we'll look at these capabilities in the next few slides. Okay. So in the context of metabolomics and exposomics, And we all know when we do untargeted analysis and to annotating them, we usually match them against libraries, or you can predict what compounds could have produced a spectra. And after you do a library match to do the identification or annotation, usually we have to do manual curation of the results as well, because no matter how advanced our library matching algorithm is, and humans still play a crucial role in curating your results. And we are trying to develop a web resource where we make it as smooth, as user-friendly as possible for this entire process. Okay, so that's what ADAP-KDB was designed for identifying and annotating compounds. So here you see a screenshot of the landing page of the ADAP-KDB web resource, and which is available at adap.cloud. Okay. So when you go to adap.cloud, you will be able to do compound identification by matching against your own in-house library or high-quality public available spectra. You can do compound annotation by matching against publicly available libraries. And you can do simple search or even a batch search. And you can visualize and interact with your compound identification and annotation results. Okay. You can also do compound prioritization, and we'll see in the next few slides what I mean by that. So for the purpose of identifying and annotating your compounds and for prioritizing your compounds, we have multiple spectral libraries and compound libraries that you could search against in ADAP-KDB. And this include consensus spectra that we have built from publicly available data. We have reference spectra that we have assembled from the public domain. And there are also compound libraries where there's no MSMS, just the mass formula, those meta information. Okay? And this collection of libraries within ADAP-KDB is constantly being updated to include more and more spectra and more compounds. So it is very dynamic. So as we add more and more spectra into ADAP-KDB, so search speed becomes an issue. So we have put quite some effort to try to speed up the library search. So here I'm showing some of the work we have done for speeding up the GCMS library search. And basically we try to do some filtering before we do to calculate the scores to make it faster. And for those of you who are interested, I did list the publication for you to look into. Okay. So that's compound identification and an annotation by searching against both private and public available spectral libraries. For compound prioritization, what do I mean by that? So for untargeted GCMS or LCMS analysis, we see so many signals that we cannot identify or annotate. I'm sure many of you have had experiences. So for those large proportion of unknown signals, we cannot afford, usually, to try to identify or annotate them all. And so in addition to try to predict them, we also try to prioritize them to see what unknown signals we should target. And this is where the compound prioritization comes into picture. The basic idea is by looking at data in the public domain, we can see what unknown signals are frequently observed across different species, across different sample sources, and across different sample diseases. And so that's where compound prioritization is for. So how do we do that? Okay. As we all know, there are multiple large-scale public available data repositories for untargeted mass spectrometry-based metabolomics and exposomics. And this includes Metabolomics Workbench, which is probably one of the largest in North America and probably in the whole world as well. In addition to that, there is Massive in UCSD, there is Metabolites in Europe, and there are some other big data repositories. The idea is we use ADP-BIG to pre-process the data in these public domains. It's a unified secondary data pre-processing. And from those untargeted data, we construct MSMS and purify GCMS spectra and cluster them, even though they're from different studies, from different even instruments. And then for those spectra that in the same cluster, and we try to provide statistics for those spectra in the same cluster. Those statistics is used to give us information about how important these unknown signals could be. And you could target your very intensive compound identification efforts for those high priority unknown signals. And this is a work in close collaboration with Shankar's group at UCSD, and his group developed Metabolomics Workbench. And for those of you who want to know more details, you can look at the publications that I listed on the slide. And in particular, if we look at these two pie charts, you can see this is the distribution of different diseases and different species for spectra in the same consensus cluster. Okay. So I hope that have given you a brief conceptual idea about what ADP-BIG is capable of and what ADAP-KDB is capable of. Next, let's try to see if we can do a hands-on exercise using ADP-BIG and ADAP-KDB. Okay, so have you downloaded ADP-BIG? Yes? Okay. So now let's install on your laptop. Okay. So on the slides, I have instructions for installing your installer either on Windows or Mac. If you have a Linux machine, I don't know how many people are using Linux machine, and there is the Linux installation packages as well. Okay. So here on this slide, I have screenshots for installing the Mac installation package. Okay. So after you have downloaded the installation package, if you double click, and you usually might get this error warning, basically. And you do need to go to your system preferences to click that open anyway, okay? And then go ahead to install your ADP installation package on your Mac. And for those of you who are using a Windows operating system, and these are the steps for you to follow to install on your Windows computer. I'll give you a few minutes to see if you can install. Hopefully it's relatively straightforward for you. Okay. So I saw one of you ask the question about whether or not ADP-BIG can take raw instrument data. Yes, it can. So I see Alex has already answered your question. You want to look at the slide? Did I get your question right? Okay. Did you encounter any issues to install? In the Google Drive, I did include the ADP-BIG project file with all the results from ADP-BIG processing. So if you run into any issues in installing ADP-BIG on your computer, for one, feel free to email us to let us know what questions you encounter. And for two, you can use the ADP-BIG project file on the Google Drive still. Hopefully that will help you later on. And today we'll still look at some of the raw data and intermediate results. Okay. Can we move on? Okay. So ADP-BIG was designed especially for large-scale analysis. So of course, the more computer cores you have, the better. The more RAM you have, the better. But as we saw earlier, ADP-BIG was able to process more than 4,000 samples in 30 hours, which is not terribly slow, I would say. And so within ADP-BIG, you have the freedom to specify the number of CPU cores to be used by ADP-BIG. And that's in the ADP-BIG preference, and we'll take a look later. For today's hands-on exercise, we will be using a set of samples from a publicly available study on metabolites. And here I have the metabolites ID there. It's MTBBLS1066. Okay, this is an untargeted metabolomic study about molecular effects and of ketogenic diet on healthy and tumor xenograft mouse models. Okay, and more detailed information can be found on the Metabolites website. Okay, and they have both reverse phase data as well as the helix data for today's exercise. Because our time is so short, we just use a subset of the samples available on the Metabolites website. And they have both positive data and negative data. We'll only be using their positive data. So we'll be using their reverse phase positive data. And again, this is a Google Drive where you can see the subset of the data for today's exercise. And here in this CSV spreadsheet, we listed the files we will be using. Okay, so everything again is on that Google Drive. Okay, so for our hands-on exercise, we will execute entire single batch LC-MS workflow. We will explore by interacting with the raw data. And we will also explore intermediate results that includes chromatograms that we have built. We'll explore the peaks that have been detected, explore the data after alignment, background removal, and normalization. Okay. So let's first get a conceptual overview about the GUI in ADP-BIG. Okay, similar to many GUI-based software tools, we have this part. You can see the samples that are involved in your project. And here, all these icons that are the different steps in a particular workflow, no matter it's GCMS or LCMS workflows. And you could explore intermediate results as well as the final results. And here, this is where you interact with your data. And this panel shows the progress of your workflow. And here shows the logging information, okay? And that logging information is very useful for you. So now let's open our ADP-BIG. So, and if you could open your ADP-BIG, could you raise your hand? Let me know how many of you is able to install. I guess about half. And okay, thank you for installing that. Let's see. Okay. And this is I cannot use this Windows machine. So because hopefully we are all on Zoom. Are you all on Zoom? Okay, good. Then I can switch to the Zoom computer. Do you see the ADP-BIG screen that I'm sharing in Zoom? Yes? Okay, good. So do you see my cursor in Zoom? Okay, so from now on, we'll just focus on Zoom, okay? So here in this panel, the project panel, you see the raw data files that we have imported into the project. For each raw data file, we could interact and explore our raw data by clicking on Input. Okay. For those of you who have installed ADP-BIG and the raw files, you can try to, actually, you know what? I actually forgot one thing. I am going to close the project so that we can start from the beginning, okay? So we want to create a brand new ADP-BIG project by either clicking on this Create a New Project. Do you see my cursor in Zoom? Okay. Or you can click this icon to create a new project. Now you have the new project window open, okay? And here you can give your project a name or to use the default name that's created by ADP-BIG. In this name, you have year, month, day, and basically minute, second, okay? And then you want to select a location for your project file. And click that, you give it a location. For example, as I'm going to put it here, okay? So here you can see the 3 workflows within ADP-BIG. workflow for processing GCMS data, single batch LCMS data, or multi-batch LCMS data. And for this exercise, we'll be doing LCMS single batch data. Okay, so I click this workflow. Okay. Next, we select the default parameters for high mass resolution data or low mass resolution data. Of course, if you have your customized parameters, you could import your customized XML file with all the processing parameters. Okay? So here I will just use the default high mass resolution data. And later on, after you have imported data, you have flexibility to re-specify those processing parameters. Okay. Next, you can see in this panel, there is a single batch and a multi-batch. And right now, we are in the process of merging these two. But at this point, those two workflows are still separate. So because this is a single batch exercise, we basically use a single batch. Okay, if you do multi-batch, you click this tab, multi-batch tab. Okay, and then we click select files, and then select the files we are going to be using. Okay, and if you have downloaded the raw files from Google Drive, you could go to your folder on your local computer and then get those samples. So here on my computer, I have those raw MZXML files. I select them and then open. Okay. You can also directly import raw files. Okay. And next, if you have the sample metadata information, you can click this Add Factors to tell ADAP-BIG what are your sample types. For example, you, of course, have study samples. And if you have quality control samples, for example, blank samples and NIST reference standard materials samples, study pool samples, those could help tremendously with data preprocessing. And you can indicate those sample types in the CSV files that you could upload. So here, I have the file stored in this factors.csv file. And earlier, we have looked at a screenshot of those sample metadata. Here, I'm going to show again to give you a better idea what kind of sample metadata information is in that CSV file. Okay. Okay. So you see here, we have a few columns. We have the injection order. This is especially important if you do multi-batch processing. The sample name, the data files, and the sample type, okay? And also, if you want to do machine learning and statistical analysis, you need to include the factors information in your study as well, okay? And this sample type indicate are these study samples and different quality control samples, okay? And this file has been imported into ADAP-BIG. And here you can see the sample type has already been indicated based on information in that CSV file here, okay? And after you have selected your files and imported your factors file or sample metadata information, you can see statistics for those samples. This is a mechanism we implemented to help users to check whether or not all the samples have been prepared as expected. So for this particular exercise, we can see there are 5 study samples, 1 black sample, and 1 pool sample based on that sample metadata CSV file we have imported. Okay? And next, you could either check or uncheck this entire workflow execution. For today's exercise, we'll just use execute entire workflow. Sometimes if you don't want to process the data, you only want to visualize your raw data. Or if you want to do every step manually, you uncheck this execute the entire workflow, okay? So let's say I will execute the entire workflow, and we'll see how fast it can process. And you could do the same on your own laptop, okay? So now you see the processing has started, and the bottom left panel shows the progress of the data processing, okay? And in the meantime, the bottom right panel shows the logging information, okay? The parameters for the processing and how many cores are being used for each step, all those information. You might see this is a little bit too much, and we are trying to simplify this logging in window right now, okay? Any questions at this point? So how many of you could launch this processing? Good, a few, good. Okay, now you see the peak detection progress bar and also the percentage of the data processing. Of course, it's an estimate, okay? Now it's running. We can go back to this slide, see if we have missed anything. Okay. So now let's look at the big screen, okay? Let the ADP-BIG run. So as I said earlier, ADP-BIG has built-in default parameters, and that's the default parameters that we expect to work relatively okay for commonly generated data. But if you need to customize ADP-BIG, this processing parameters window is where you need to go to, okay? So here, you can see the processing parameter specification window, and for every step, when you click it, you could see the parameters you could specify, okay? So for peak detection, for example, and ADP-BIG uses wavelet transform-based approach, you can see those relevant parameters for for peak detection, and similarly for other steps. And I have the same experience or challenges for specifying parameters. And we do know it's not fully solved right now in the community, even though there are software tools to try to find the best set of parameters for you. For example, the IPO software tools and other software tools. And ADP-BIG is not the exception. We are still working on trying to relieve some of the burdens on users' shoulder. Okay? And the parameters are stored in XML files. Okay? And as we saw earlier, if you have a set of parameters that you are happy with, and you could save them in an XML file for future use, actually you could export the ideal parameter settings from ADP-BIG into an XML file, and then import it back into ADP-BIG. Okay, on my computer, if we come back to Zoom, it's already done. I guess for those 5 study samples, and 2 quality control samples, a total of 7 samples, it took a few minutes to finish. Okay, so now let's explore our raw data. Okay, so for each raw data sample, if you click, and you can see 4 clickable things, okay, underneath each sample. If you click input, this is where you can explore with the raw data, okay? So for this first particular sample, when I clicked input, you can see the list of raw spectra. For example, the first scan, you can see the scan number, MS level, polarity, retention time, precursor retention time, and so on, okay? And then the second to the 6 scans, those are the MS spectra. Again, this is from metabolites. And then after that, which is another MS1. For each scan, no matter it's MS1 or MS2, when you click, you see the raw spectrum. Many software tools have these capabilities. For example, MZMine, which I love, MS-Dial, which I love as well, and to help you to explore your raw data, okay? And in addition to interacting with your raw spectra, you can also look at the chromatogram-related information. For example, in this tab, you can see the base ion chromatogram. You can see the total ion chromatogram. You can see the retention time versus homogeneity map, okay? If I come back to the total ion chromatogram, you can see there is this filter tab. If I click, that allows you to specify a particular homogeneity range and retention time range to explore more details of your data, okay? And for each visualization, no matter it's a spectrum or chromatogram, you could zoom in by doing this. And for the chromatogram, it's the same. Or you can zoom out by clicking the home button. Okay? This is slightly different from how MZMine allows you to zoom in and zoom out. But it works for ADAP-BIG, okay? So that's the raw data. Because now the processing has already finished, we could look at the intermediate results as well. When you click Chromatogram Builder, you could look at those chromatograms, extracted ion chromatograms that have been constructed one by one. Okay? For each chromatogram, you can see some more information for them. Okay? And for peak detection, the same. Okay, what are the peaks that have been detected? And if you have MS-MS, so ADAP-BIG is able to process either just LC-MS data without MS-MS or LC-MS data with MS-MS. Of course, when you have MS-MS, when you have many samples, and then ADAP-BIG has a pairing step to link MS1 peak with MS-MS spectra. Okay? And you can do the data exploration for each sample the same way that we have seen so far. Yes? Yes?
So when you say pairing of MS1 and MS2, what do you mean?
Basically, after you have, especially after you have aligned many samples together, what are the MS-MS for a particular MS1 peak in your peak table? Does that make sense? Yes?
So here we have MS1 and MS2 data, right?
Yes? Uh-huh.
So in the output, there is then the new list with MS1 and MS2 spectra.
Mm-hmm. Yeah, yeah. And also, sometimes if you do DDA, even though when you have dynamic exclusion window specified when you acquire the data, sometimes you still might have multiple MS-MS for the same MS1 peak. In this pairing step, we also try to filter out some of the MS-MS. Does that make sense?
Yes. So is that can you export it again?
Yes, yes, yes. We'll see that step later on, okay? That's a good question. Honey?
I was going to ask how that filtering of MS-MS
Very good question. So right now, we basically, when we have multiple MSMS for the same peak, and we try to choose that MSMS that's closest in retention time to the peak center of the MS1 peak. Right. Yes. Any other questions? Yes? Right now, it's primarily for data-dependent acquisition. Good question. Yes?
Yeah, as you said, so in future, it might be you can add multiple MSMS to one MS kind of for a line.
We do want to have the capability to just keep one MSMS for MS1 peak, or if you want to keep multiple, we do have a step to cluster the MSMS for each peak and keep them all. Does that make sense to you?
Yeah, as you said, the stacking to the retention time. What about the mass accuracy? You said any parameter like to 1 ppm, you are accommodating this, or within your range versus 2 ppm?
Very good question. Nowadays, mass spec used for acquiring LC-MS/MS data usually have very good mass measurement accuracy, like 3 ppm or 5 ppm. We haven't found the mass measurement, the immobility tolerance makes a big difference because the mass spec is already so good. It's a retention time we find that's could play a role. Does that answer your question? Right. When we link them, we use them both. But retention time is the parameter that if we want to just keep one MSMS for that peak. Yes? Any other questions? I'm very impressed. You guys are asking really good questions. Yeah. To tell the truth, before I stepped into this room, I was worried maybe there are only less than 10 people in this room who will join. This is the last minute thing. This workshop was originally not selected, actually. And I'm very glad that we could showcase ADAP-BIG to you. And thank you again for joining today. Okay, without further questions, we'll continue. Okay. So now we have seen we could interact with raw data and intermediate results. Now let's look at the data after alignment, right? So if you click the within batch alignment, remember this exercise, small project, processing project, has only one analytical batch. So we only have within batch alignment. So here, after you have clicked this within batch alignment, you see the list of peaks that have been aligned. You see here? So for every peak, because we have 7 samples, including 5 study samples and 2 quality control samples, so you see 2 visualization panels. The top right panel here shows this intensity versus sample, okay? That'll give you an idea how the intensity changes across different samples, okay? And you can also look at the retention time fluctuation for this particular peak, the fluctuation of retention time across the samples. And ADAP-BIG also give you a quantitative idea by showing you the absolute difference of retention time of that peak across the different samples. So basically, we try to make it easy for you to explore the intermediate results after alignment. Okay. Yes?
Before the alignment, can we exclude any bad acquired data? Or we have to see after the alignment?
Good question.
Quality, and can we have the option to exclude?
Unfortunately, no, but I'm going to double check with Alex. We could add new samples, but to exclude some samples, you might have to recreate a new project. Alex, are you there? If you don't mind, you could type your question in the chat box. I don't think ADP-BIG can, unless Alex has implemented something that I don't know yet. Okay, good question. But I think it's needed that we have that capability to exclude certain samples. Yes?
So you explained it to use. So how do you typically use that then in assessing if everything went well, or what's the function?
Oh, if you have quality control samples, for example, blank samples or study pools, after the alignment, we have a principal component analysis step to see whether or not it's two things. Whether or not your study pools are basically somewhere in the middle of your study samples. That's one way we check. Another thing we check is whether or not if you have blank samples, the blank samples are quite distant from your study samples. Does that answer your question?
Yeah, so PCA you don't show here, or do you? PCA, I've seen the two views that you show are not the PCA, right?
On the slides, actually, I did include the PCAs here. And you can do principal component analysis or some other supervised classification approaches as well.
Okay. But my question was more to the two screens you showed that we also see after clicking on the within batch alignment feature.
Mm-hmm.
The variation of the intensity and of the retention time. So how do you typically use that? Just to check how well the alignment went? Or?
The idea behind that visualization really to see whether or not your retention time tolerance, which is a parameter in alignment, whether or not it is appropriate, and also see for different peaks how much the peak intensity change across different sample groups. For example, if you have control group, you have based on your factors file.
Okay.
Make sense to you?
Yeah.
Okay. Yeah. Honey, do you have a question? No? Okay.
I have a question. So this is a single batch, and is there a limit number of samples can be got? Like what's the maximum number of samples?
Oh. We have not tested one analytical batch with thousands of samples, but a few hundred samples should be no problem at all. But usually when you have more than a few hundred, you have multi-batches, right? Yeah. Any other questions? No? Okay, good. So let's continue. And in this version that you see, you actually don't see the principal component analysis included in this. We did not include it in this particular release. Actually, after alignment, after background removal, after normalization, we have the PCA plot at each step to help you to see whether or not your sample data has been acquired properly. We do use those quality control plots to identify possible issues with samples. For example, samples have not been prepared properly, or samples have not been acquired properly, and we use that plots as a diagnostic mechanism to help analytical chemists to identify issues. Okay?
So do you do that automated or what? Yeah, like visual checking of?
It's automated. In our design, basically, after alignment, after background removal, after normalization, PCA is automatically calculated and plotted, which is not in this release, but hopefully next release it's going to be there. Make sense? Okay. Yeah.
I'm kind of curious to how you call it PCI. We got this.
Good. And later on, you will see after the data processing, ADAP-BIG will export a PDF report. In that PDF report, we automatically include the PCA plots to help basically people to check whether or not the processing has been done properly or the samples have been acquired properly. The PCA plots, again, are used as a diagnostic plots for identifying problems. Okay. So next, I think we have gone through those. Okay. So after the processing has finished, we could export two things. One is your CSV peak table, okay, which is here. Export your semi-quantitative peak table into a CSV file. We can also export your MSMS if you have LC-MSMS data into an MSP file for subsequent compound identification and annotation. So for this one, you see, I'm going to export, and you can do the same if you have run the entire workflow. And then I can give it a name, for example, peak table. Save. I already have it, so I'll just replace. And for the MSMS that have been paired with MS1 peak, you can click that Export to MSP button and then select Save to File. Okay, so here I have the spectra in MSP. And another possibility is to send your MSP directly to ADAP-KDB, and which we are still working on that. Okay, for this release, we will not look into that. So I have saved our MSMS into MSP file on my local computer, right? Okay, so next, we need to try to identify or annotate those MSMS to see what they are. And in order to do that, we'll upload those into ADAP-KDB for compound identification and annotation. And we can go to the next slide. Oh, I forgot one thing. As I said earlier, you could produce a processing report in PDF format by going here, export processing report to get a PDF file, okay? And that's shown here. In this PDF report, you can see the basic information about the project, what's the project name, the location, what's the ADAP-BIG version, and the workflow that you have selected, and also some general information about the peaks, number of peaks you have detected, number of MSMS that you have exported into your MSP file, and the PCA plot, all those. And we specifically generate this report so that we could have a dedicated person to double quality check our results before we deliver our results to our clients. Okay. So far we have looked at using ADAP-BIG as a GUI application, you could also deploy ADAP-BIG on a computer cluster if you need faster processing by using more computer cores. And I'm showing here basically the setup for you to do that. If you have any questions when you try to do that, just feel free to let us know. On the ADP GUI, you could email us if you have any questions. Okay. Next, we'll try to identify and annotate the signals in our MSP file. And for that, we'll go to our cloud resource, ADAP-KDB. In a nutshell, for identifying and annotating compounds, we could do that in ADAP-KDB by uploading the MSP file that ADAP-BIG has produced for you, select the libraries that you want to search against. That library includes your own in-house library if you trust ADAP-KDB, and upload your in-house library into ADAP-KDB, or we can search against publicly available spectral libraries. And after the search, we could export those search results, of course. So let's look a little bit more into detail while I run this. So we'll look at Zoom again and do our hands-on exercise. Okay. So the ADAP-KDB website is at adap.cloud. Okay, and you can go there, adap.cloud. So in order to upload our files, we can click this Upload Files button. Okay, I'm going to clear an analysis that had been uploaded. So now I don't have any data. Okay, I'll upload the file by selecting what are the spectra we are going to use as a query spectra. So for this exercise, it's LC-MS-MS plus. It's positive polarity. And then we click Choose Files. Then we select our MSP files that we have exported from ADAP-BIG. So here is the MS file that had been produced by ADAP-BIG. Okay. So I have selected, and then just check I'm not a robot. Okay. And then upload. Okay. So here we have the field mapping capability to let ADAP-KDB know what are the fields that ADAP has produced in the MSP file. So let's open that MSP file that have been produced so that we understand what we are dealing with. Okay, so this is the MSP file that ADAP-BIG has produced for us. And I assume most of you are very familiar with MSP format, which is quite similar to MGF format. It's a text format for storing MS information, right? So in this particular MSP file, we have multiple fields. We have name, comment, DB number, retention time, and so on. So after this MSP file has been imported into ADAP-KDB, those fields are listed on the left-hand side. Do you see in Zoom? Okay. And we need to let ADAP-KDB know what are these different fields. Okay, what are the fields that you should care when you search those query spectra against libraries. And you can drag and drop particular fields into here. For example, the name field in MSP file is name. You can just drag there. I could even drag them back. Oh no, I can't. Okay, and so the name, I think there is a bug here. And then the precursor member Z should be recognized as precursor member Z, okay? So here, the right panel is the read as panel. Read each field in MSP file as a particular field that ADAP-KDB can recognize. And read the retention time in MSP file as the retention time, okay? If those fields needs to be used for library match. So here for this particular exercise, we will use 3 fields. The name field as a unique identifier for each MS spectrum, the precursor number Z, and the retention time, okay? And then we just click Add Metadata, okay? So now you can see every spectrum in your MSP file has been imported into ADAP-KDB, and you can click each of them and see that spectrum. Here you see the metadata for that spectrum. Here you see a plot of that spectrum. And next, we could do a library search by clicking on this search button. ADAP-KDB has 3 search modes. You can search your query spectra for similar spectra in ADAP-KDB library. You could search for similar spectra and also request ADAP-KDB to assign a confidence level for your library match. That's an ontology level. And you can also search query spectra for similar spectra that have been appeared in different studies, and this is for compound prioritization purpose. At this point, let's just focus on search for similar spectra, okay? So if I click search for similar spectra, and here you could specify what are the libraries you want to search against. To make the search fast for this particular exercise, I'm just selecting the mass spec release from last year. You can also uncheck other libraries, okay? So, and then you can specify search parameters, and you can also give ADAP-KDB an email address so that you don't have to wait here. After the search is done, ADAP-KDB will send an email to you, let you know it's done. You can come back and check the library searching results, okay? So I'm going to use the default parameters, use a mass spec release from last year to do the search. So now, after I have launched the library search, here you see the progress bar and also the matching results underneath, okay? And this panel, the query signals, is a list of signals or list of MSMS in your MSP file. And for each signal, you could see the match results against the library. Here is the mass spec release 2022. And you also see if there is a good match, you see the structure of the compounds in the library. So you could click through this and visually reveal the matching results after and during the search. Okay? Any questions at this point? Yes?
Yeah, if I click on a random spectrum, it does have metadata. There are no tubes.
Did you upload the MSP file or the CSV file?
MSP file.
You don't see the raw spectra?
Yeah, there are no tubes currently, but I can check in the MSP.
Can you open the MSP file to see whether or not there are peaks in that spectrum?
Yeah, I will. So it does include empty spectra there as well?
It should not. If it's empty, it should not be even exported into the MSP file.
Yeah, there are peaks in there. So
It was just not displayed.
okay.
Do you mind sending us the bug? or you can just send the MSP file to us so that we can take a look why the spectrum has peaks but is not displayed in ADP-KDB. That will help us identify the issue. Good, thank you. Okay. So after the search has finished, you could export your search results by clicking on Export. And then here, there are two options. You can export your search results into a simple CSV file. That's a simple export. You can also export your search results into an advanced report. The difference between these two is sometimes you might have multiple hits for each query spectra, and advanced export will include everything. Okay? So if I click the simple export, for example, it will produce this Excel file. Okay, so I just opened the Excel file. If you can look at Zoom right now, if you can still see my screen, in this exported search result Excel file, there are two worksheets. One worksheet is an index worksheet. It's basically a data dictionary to explain the different columns in that data worksheet. Okay, so in this worksheet, you can see the search results are divided into multiple, actually here is 3 or 4 color-coded sections. You have the light blue, that's a query signal section. You have the yellow, that's a library matching some metadata, and then the compound information is in the green section. Okay, so that's a compound identification and annotation in a nutshell. Okay. Any questions? No? Good. So next we will keep going. I think we have done this, done this, done this. Okay. Search against private library. As I said earlier, if you trust ADP-KDB and you want to use ADP-KDB as a search engine for your private library, ADP-KDB does allow you to create your own private accounts. And then you can upload your in-house library to ADP-KDB and indicate it's private. No one else can access. And then you can upload and then search against your in-house library. If you have multiple people in your lab or institution that need to share the same in-house library, you could create an organization on ADP-KDB, and all the people in your organization would share the same in-house library. So ADP-KDB has all those capabilities. Okay. So next, I want to spend a little bit of time on ADP-KDB's capability for you to prioritize signals, especially those unknown signals, by looking into statistics of similar spectra in public domain. Okay? So here, if you look at this slide on the big screen, and that's the consensus spectral statistics. If we come back to Zoom, I'm sorry, we're jumping between the two. Okay. If we come to ADP-KDB again, if you click on the Libraries tab on the left, you can see View All Consensus Spectra, as well as View All Public Studies. If you click on the left, View All Consensus Spectra, right now, the library contains about 47,000 spectra from the public domain. And for each spectrum, ADAP-KDB provides the meta information about where the study is from. And those spectra, because they have been clustered, you can see the spectral similarity for different clusters, and also the ANOVA p-values to help you to prioritize. And in addition to that, there is also the goodness of fit value also to help you, the p-value to help you to prioritize. So let's see if I click on the studies column. Okay, let's see a consensus spectra that have been observed repeatedly in different studies. For example, this one. It has been observed in 33 studies, similar spectra. And here ADAP-KDB shows a pie chart to display the percentage of samples or sample sources or diseases where similar spectra have been observed, as well as the diversity, which means how diverse these different pieces of metadata is to help you to determine whether or not this is worth your efforts and funding to try to do compound identification using experimental methods. So that's the idea behind ADAP-KDB signal prioritization. In a nutshell, it uses consensus spectra and frequency of observations and also statistical significance to try to determine whether or not it's worth the effort to go through the sophisticated experimental compound identification method to identify those compounds. Okay? So all the exercise data that we have gone through is available on these links. And ADAP-BIG and ADAP-KDB, of course, is open source and freely available to everyone in the community. And we do welcome feedback from each of you because I consider our mission is to make your life as easy as possible. So anytime when we receive a feedback, we take it very seriously and try to make ADP informatics framework as useful as possible to the community. So now with only 20 minutes left, I would like to know whether or not you have any questions for me. So now it's a Q&A session. Honey? Say that again, honey? Yes, we are working on, let's see, I think three manuscripts. One is ADAP-BIG algorithm, the workflow for big data. One is ADAP software, where we explain how everything is optimized to allow ADAP-BIG to process big data. Another is to compare ADAP-BIG against two commercial software tools. One is Progenesis, one is Compound Discoverer. So our comparison results so far have indicated it's at least comparable, but the big difference is Progenesis and Compound Discoverer, as I know so far, takes a lot of time or even incapable of processing studies where you have thousands and thousands of samples. Thank you. Yes, Aparna? Right. We have user manual for ADAP-BIG that's already on the ADAP-BIG GitHub website. And for ADAP-KDB, we have a simplified user manual for GC, we are working on a very detailed user manual for everything else right now. And regarding a video tutorial, I'm hoping that we could start recording videos and put them on YouTube. That's something we are working on. Yeah. Yes?
So you mentioned the commercial software. There's also MSFLine, MS-Dial, a number of other initiatives. So are you also planning to give some insight on how you think, ADAP-BIG fits into that kind of ecosystem of tools, so to speak?
Right. We have done detailed comparison of ADP-BIG against MS-Dial in our past publications. We have compared against XCMS in the past publications, Analyzer Pro for GC, and peak detection for both GC and LC. And as we said earlier, ADP-BIG is also available in MZMine. And within MZMine, the peak detection, it has a synthwave from original XCMS. It has ADP-BIG, the ADP peak detection, basically. So in the past, we have done quite a lot of comparison with these publicly available open source software tools. So recently, we tried to do a comparison, a very detailed comparison with commercial software tools, and that is Progenesis and Compound Discoverer. One represents waters, one represents.
I was just wondering how you see that, okay, let's say that there are some MSFLine users in the room, right?
Uh-huh.
So in which case should they use, I mean, are there special cases where you think, okay, here ADAP-BIG can be really of benefit over MSFLine, or because in MSFLine you can also do live research, you get mirror plots, I mean, so that's why I was wondering, like, hey, how do you see where your tool fits in? I mean, of course, you can compare it on how fast it is, but yeah, I'm more curious to hear your view on when you think ADAP-BIG can make a big difference.
Okay, I can answer your question from two perspectives. Okay, one is ADAP is already part of MZMine. If you look at Google Analytics for chromatogram building, and many users of MZMine actually use ADAP chromatogram building and peak detection. And ADAP actually was originally incorporated into MZMine way before we started developing ADAP-BIG. But ADAP MZMine was not designed a few years ago. Fundamentally, it was not designed for processing big data. That's why we started developing ADP-BIG, its own software too. So in terms of where ADP-BIG fits in this landscape, the number one is if you have thousands and thousands of samples as you need to process.
Yeah. Although I think they are quickly catching up, but that's fine.
Right, right. I have been communicating with Thomas because we are still maintaining ADP-BIG within MZMI. I actually told him in person in 2019, I said, we need to process big data, and MZMine just does not allow us to do that. We recently started developing ADP-BIG so that we could process the data that's coming to us.
No, it makes sense.
Yeah.
But now, at some point, you run the risk that you have two ecosystems that perform similar tasks, and then it makes sense to appreciate whether to use which one.
Right, right, yeah. But ADP-BIG does not have the capability that MZMine has to process eye mobility data, DDA data. I think MZMine has become so much more comprehensive than ADP-BIG. In our work, we have projects coming our way from funding agency, funded projects to process lots of DDA data. And so ADP-BIG has been developed specifically to deliver results for our clients in that arena. Okay.
I just have one suggestion. Maybe you already have this feature. Usually, to avoid human error, it would be better to import it where data sheet instead of keying those covariant. And we have those metadata sheets that we can export. Do you have this feature to export from the either text or CSV file? And so that to avoid another human key for simple by simple when you start the project.
You mean import the metadata? You can, for single batch, you can manually specify on ADP-BIG or that sample type. And I found that is more error prone. Or you can create a sample metadata CSV file with all the sample metadata and then load that into ADAP-KDB. Is that your question? Okay. I prefer a CSV file instead of manually specify the sample type on the GUI. I feel that's more error prone, but we do have both capabilities. And for multi-batch, you have to have the CSV file. When you have thousands of samples, it becomes utterly unrealistic to manually specify the sample type of every file, right? So you have to have a CSV file. Okay. So the community has been working very hard in creating informatics capabilities to help our metabolomics and exposomics community. For some of you I know, like Hani, I know you have developed binner and metabocombiner and done a lot. And the gentleman, I forgot his name. I know his lab has done tremendous amount of work as well. So bioinformaticians like us, our sole goal is to help analytical chemists, experimentalists, and people who run a core. I know you run a core, to try to make your life easy. We collaborate closely with multiple labs core facilities who generate the data, and they want to have informatics software too that could relieve some of the burdens on their shoulder. And we are fully determined to do that, even though we have been doing that for years. So again, if you have any requests for new features, if you find any bugs, please don't hesitate to let us know. And if you need some help with looking into the data you generate, feel free to get in touch with us as well if you need new features. So that's basically what we do. Yeah. Okay. And also the source code is open. Many of the source codes are on GitHub, and you are welcome to use some of the source codes. And we'll be happy to help you to incorporate into your own software tools if you need. Okay. Thank you. Thank you. A big shout out should go to Alex, who is on Zoom, who has led our software team of software programmers in developing all these capabilities. Alex, are you there? I cannot see him. Okay, Thank you for all the hard work, Alex. You see him? He's a handsome gentleman, very handsome. I feel he's like a movie star, his look. And he works extremely hard as well. Everyone just loves him, including me. So thank you all. Yeah, enjoy the rest of the conference. And thank you again for coming. Thank you for all the questions, most of all.